Good morning, students. Today's topic is immunity. We know that every day we are exposed to a large number of infectious agents. They may be bacteria, virus, pathogens, protozoans, helminths, etc. Not every time we get the disease. Why is it so? This is because our body is able to defend against many of the microbes that are entering our body. First of all, our body tries to stop the pathogens from entering our body. But if they fail to stop and pathogens accidentally enter the body, then also our body consists various mechanisms by which we can kill that pathogen. That is, the body is able to defend itself from most of these foreign agents. This overall ability of the host to fight the disease causing organisms conferred by the immune system, that is, the body consists immune system, and this is known as immunity. When a foreign particle enters the body, the macrophages engulf them, and after lysis they represent their parts as antigen. Now these cells which are representing the antigen are known as antigen presenting cells. Immunity is of two types: innate immunity and acquired immunity. Innate immunity, that is non-specific. It is present at the time of the birth. It provides different types of barriers like skin, mucus, acid in the stomach, and so on. The barrier prevents the entry of the antigens into the body. Another one is acquired immunity, that is also known as pathogenic specific immunity. Its characteristic feature is memory. It consists of primary and secondary responses. Our body memorizes the pathogen from its first encounter and reacts accordingly during second encounter. This describes the fact that our body appears to have memory of the encounter. The primary and secondary responses are carried out by T lymphocytes and B lymphocytes. Innate immunity consists of four types of barriers. They are physical barriers, that is, the skin on our body is the main barrier which prevents the entry of the microorganisms. Another one is mucus coating of the epithelium lining in the respiratory tract, gastrointestinal tract, and urogenital tract also help in trapping the microbes that enter our body. Another one is physiological barriers, that is acid in the stomach, saliva in the mouth, tear from the eyes, all prevent microbial growth. Very low pH cannot support the microbial growth. Similarly, lysozymes in saliva and tears also act on the microbes and prevent the microbial growth. Third one is cellular barriers. Certain types of leukocytes, that is WBCs, or of our body like polymorphonuclear leukocytes, that is PMNL, known as neutrophils, and monocytes, along with the natural killer, that is a type of lymphocyte in the blood, as well as macrophages in between the tissues, can phagocytose and destroy microbes. Next is cytokine barriers. Virus infected cells secrete protein known as interferons, which protect non infected cells from further viral infection. So, these are the barriers which are the lines of defense in our body, which are covered under the topic innate immunity. Now, the next is acquired immunity. On the other hand, it is pathogen specific. Unlike of the innate that was non-specific, here it is pathogen specific. That is, it will first identify the pathogen, that whether it is self or non-self, and accordingly only it will create a response. So, it is characterized by memory. When our body encounters the pathogen for the first time, a very low intensity response is produced known as primary response. On the other hand, subsequent encounter with the same pathogen elicits a highly intensified secondary response, also known as anamnestic response. This describes the fact that our body appears to have memory of the first encounter. The primary and the secondary immune responses are carried out with the two types of special lymphocytes present in our blood, that are B lymphocytes and T lymphocytes. B lymphocytes produce an army of protein in response to pathogen in our body to fight with them. These proteins are antibodies. The T cells themselves do not secrete any antibody, but they help the B cells produce them. Structure of antibody. Each antibody molecule has four peptide chains. Two of them are small, known as light chains, while the another two are heavy chains. Hence, antibody is represented as H2L2. Heavy chain consists of variable region along with the constant region. Similarly, light chain also consists of variable region and constant region. We can see here that heavy chain and light chain forms the structure of antibody. Constant region is represented by blue color while variable with red. The variable region consists of antigen binding site while constant region consists of Fc region, which recognizes the cell. Antibody may be a single molecule as in IgD, IgE, IgG, while double in IgA and many in IgM. We can see the types of antibodies over here: IgG, IgE, IgA and IgM. On the base of response, acquired immune response can be of two types, humoral and cell mediated. Since the antibodies are released in the blood, which is flowing inside the whole body, the response is known as humoral immune response, while when the cells like T lymphocytes are involved, the response is known as cell mediated immune response. We can see both the responses via a animated movie. Here is the antigen presenting cells. T helper cell will identify them. There will be some chemical release from these cells which will ultimately goes and stimulates the B lymphocytes. These B lymphocytes starts producing antibodies while the cytotoxic T cells engulfs them. Now the infected cell, which is representing the antigen, will be encountered by the cytotoxic T cell. When the cytotoxic T cell encounters the infected cell, it releases the chemicals which will cause the death of the infected cell. Similarly, B cells in the humoral response will identify by the T helper cells and starts dividing and differentiating into plasma cells which will produce antibodies in the blood. These antibodies will bind to the antigen engulfed by the macrophages and will represent the antigen. This way, the antibodies help the macrophages identify the foreign agent. Along with this, B lymphocytes are also differentiated into memory B cells, which will memorize the antigen and will help in producing secondary response. Very often, when some human organs like heart, eye, liver, kidney fail to function satisfactorily, transplantation is the only solution remedy to enable the patient to live a normal life. Then the search begins to find a suitable donor. Who is the suitable donor? What a doctor should check in the suitable donor so that the transplantation may be the successful event. It is not that graft from just any source, that is animal, another primate or any other human being, can be made, since graft would be rejected sooner or later. So, for the acceptance of the graft, it is must that tissues should be matched, blood groups should be matched before taking any graft. And also, even after this transplantation, patient has to take immunosuppressants all his or her life. This way, we have seen that the body is able to differentiate between self and non self, and the cell mediated immune response is responsible for the graft rejection. These are the parameters of the immunity we have discussed here. Another some parameters like active passive immunity, vaccination, immunization, and allergies, autoimmunity, we will discuss in the next lecture. So till then, keep studying.